Hello students, this is the video which will help you to write the experiment number 1, isolation of DNA from plant material in your practical notebooks. First write the objective of the experiment, that is to isolate DNA from available plant material such as spinach leaves, green pea seeds, papaya, banana, strawberries, etc. I suggest you to use strawberry to isolate DNA, as out of all plant material, best results come with strawberry. Next write the requirements. We require plant material, जो भी हमको चाहिए, then mortar and pestle, beakers, test tubes. We require distilled water, liquid soap, table salt, gauze or any stainer, chilled ethanol and toothpick or skewer. Principle behind the experiment. DNA is found in cells of every living organisms and it is the blueprint of life, but it is incredibly small. We can see DNA by extracting and isolating chains of it. The isolation of DNA begins with lysis of cells. The soap solution breaks up the fats and proteins that are the part of membranes surrounding the cells and nucleus. Once these membranes are broken, DNA is released from cells. The salt which we add enables DNA strand to come together. The DNA precipitates out of solution when chilled ethanol is added. The ethanol separates DNA from other components of cell. Write the procedure used to extract DNA. Step 1. Take plant material. Here we are taking 2 strawberries in mortar and add half cup of distilled water. इसको मैश कीजिए to make slurry. Step 2. Add 1 tablespoon of liquid soap and 2 pinches of salt and continue mixing and mashing the slurry. Step 3. Filter the mixture by passing it through gauze to remove solid parts of strawberries. Step 4. Take small amount of filtrate in test tube and add chilled ethanol. Take care that 2 liquids do not get mixed up. Step 5. Observe the line between the strawberries mixture and ethanol. Step 6. Take out the DNA strands by using toothpick or skewer. Next, draw stepwise diagrams as shown here on left side plain sheet of your practical notebook. Use sharp pencil and label all parts carefully. What have you observed? Write it in observations. DNA appears as white precipitates of very fine threads on the spool. And in the end, write precautions to be taken. First, all the glasswares used must be thoroughly cleaned and dried. Second, chemicals used for the experiment must be of standard quality. Third, vigorous shaking of mixture should be avoided. And fourth, ethanol should be chilled. So, that's all for today. Thank you very much for staying with me. Subscribe the channel to get the information about my new video. Thank you so much.